So we're all here because Frederick Twart discovered phage in 1915. But what I want to tell you about is the interactions between bacteria and animals, which is, and one important discovery that was made was in 1934. And this is seemingly unrelated to phage, but Claude Zobel first discovered that bacteria can actually induce the development of animals, of marine animals specifically. And the way this works is that you have a bottom-dwelling marine animal, and these can be corals, urchins, tube worms. They reproduce forming free-swimming larvae, and these larvae swim around the water column, and then at some point they'll metamorphose back into the adult form. In many cases, these larvae require a cue that tells them this is a good spot to metamorphose because it's a giant ocean out there, and they need to metamorphose in the right spot, otherwise they'll be dead. If they don't have the right nutrients and there are predators around, they won't be able to grow up to sexual maturity. And so what Zobel found was that bacteria are actually this cue for metamorphosis in many cases. And so this has important implications in terms of symbiosis between bacteria and animals. This is a case in which bacteria are actually mediating a very dramatic development of many different animals in the environment. It's important for animal ecology. You can imagine that the distribution of bacteria in the ocean might dictate the distribution of animals in the environment. And it also has to do with phage, and I'll tell you why in a second. All right, so in 1962, Kageyama and Agami found things that looked like phage, but they were headless. So they're essentially headless phages. And the way that works is that in contrast to typical bacteriophage, which attack bacteria and then reproduce and lyse the bacterial cell and then release these particles that go on to infect new hosts, phage tail-like bacteriocins are produced by a bacterial cell. They lyse, releasing these particles, and then they go on to attack other bacteria. So they're essentially used as weapons by some bacteria to attack and kill other bacteria. And then we'll skip ahead quite far. In 2004, something really cool was discovered by Mark Hurst and colleagues, which is that phage tail-like bacteriocins could actually interact with animals, in addition to being able to interact with bacteria. In this specific instance, the bacteriocins were able to cause virulence in grass grubs. And I think this is quite a monumental discovery because this allows us, our thinking to expand in terms of phage that can actually, or phage tail-like structures that can both interact with bacteria and in animals in that case. So we can add to our figure phage tail-like bacteriocins that interact with bacteria and then also ones that interact with animals in virulent ways. And then in 2014, what we found was that bacteria can use these bacteriocins in both virulent ways, such as Mark Hurst found, but also in beneficial ways. And one thing that was cool about these bacteriocins was that they formed multi-tailed arrays. And so this particular multi-tailed array here is formed about 100 phage tails. They're held together by tail fibers. So in this cross-section cryotomogram, you can see the tail fibers actually link the different tails together. These are contractile, and so they contract and eject out that inner tube. And this whole structure is held together by a hexagonal lattice. And so this entire lattice holds this whole structure together. And they actually expand once the cell lyses and releases these things. And what was really cool about these things is that they actually mediate the metamorphosis of a marine tube worm. So these multi-tailed structures were important for inducing the metamorphosis of this tube worm larva into the adult form. Specifically, this tube worm is called Hydroides elegans. It's a model tube worm for biofouling and developmental research. And what's cool about these larvae is that they actually require bacteria in order to metamorphose. And what I mean by that is if you grow these larvae in the laboratory and you keep their glassware clean and their culture water clean, they'll actually never metamorphose into the adult unless you expose them to a bacterial biofilm, suggesting that they require bacteria in order to complete their life cycle. So that's really cool that, and what we think is going on here is that these phage tail-like structures are actually mediating that developmental process. Okay, and so in addition to bacteriocins that interact with bacteria, they can also interact with animals in virulent and beneficial ways. So what do I think the future of phage tail-like structures that interact with animals looks like? And I think it looks really cool. There are things that we can think about in terms of interaction, like bacteriophages that need to interact with their hosts. They probably have specific mechanisms in order to specify their specific targets. We also know from type 6 secretion systems that these inner tubes and the tail spikes might carry cargo that is delivered to the animals and have some effect by causing virulence or beneficial functions to their animal hosts. And we also might think that in the terms of these multi-tailed arrays, there might be cooperativity between phage tails. So perhaps if one is triggered, adjacent phage tails are triggered and fire at the same time to increase the payload that's delivered, or increase the distance by which these things can, the working distance by which these things can use to hit their animal target. Okay, and then once we figure all these things out, because probably in the next 20 or 30 years, these things will be figured out. But we could then use these things for the benefit of people in the environment. So we could imagine using them to inhibit biofouling, or the accumulation of animals on submerged surfaces, such as ship hulls. These animals are a big annoyance to places like the US Navy, which causes increased drag and fuel consumption of their naval ships. We can also imagine that this could possibly facilitate aquaculture. Some of these animal species are unable to be cultured in the laboratory, possibly because they can't be induced to metamorphose. And we can also think, like phage therapy, maybe these things could be involved in therapeutics, but not against bacteria, but perhaps to deliver therapeutics to animal cells or cells within you and me. I think the future is wide open in that sense. All right, we can also think about diversity, and from the few things that we know about these phage tail-like structures, there's a hint that these things are very diverse and in a lot of different environments. So this is a reconstruction of the phage T4 gene product 19 phylogeny, and you can see that tube domains are derived from both bacteria and from phage and bacteria. And the colors indicate the bacterial species in which these genes are found. And you can see that they're found in many different types of bacteria, such as cyanos, Bacteroides, gamma proteobacteria, and even in archaea in some cases. What we also know right now is that they interact with a wide variety of hosts, both in the marine environment, so in tube worms, and also in terrestrial environments like grass grubs. Some of these structures are found in endosymbionts of wasps. And some of those Bacteroides species were actually isolated from the human gut, and they carry these gene clusters that are involved in possibly interacting with animals. And so how they're doing that, we don't know, but I think the future is wide open to discover how those things are working possibly in our gut. Okay, and the last thing I'd like to talk about is animal development and animal life history. And the thing I know best is how bacteriocins might be important for interacting with animals in terms of their metamorphosis. And so that's just one facet of development. But here I'm showing a phylogenetic tree of the animal kingdom. And what I've done here is I've put a check mark next to those taxa in which there are species that metamorphose. And you can see that this spans the range of bacterial, or the range of animals within the entire animal kingdom. What I've also done now is highlight those taxa in which there are species which are induced to metamorphose by bacteria. And you can see that this is actually also spans the range of animal species. Right now our models for metamorphosis are Drosophila and Xenopus. However, these are only a very few specific model systems which are actually not induced to metamorphose by bacteria. Placing Hydroides elegans is a really nice model to study how this interaction takes place, possibly in the environment mediated by phage tail-like structures. Okay, and then we can also think about how these things might be important for animal ecology. So do phage tail-like structures and the distribution in the ocean dictate the distribution of animals in the environment? We could ask the question, if these phage tail-like structures weren't around in the environment, would there be coral reefs even? We also might think that in addition to mediating animal development, such as metamorphosis, perhaps they also interact and mediate animal development, or mediate animal behavior. We don't really know. And also, on the flip side, do they mediate animal ecology, or sorry, bacterial ecology? These bacteriocins are being produced by a subpopulation of the bacteria within a biofilm, and they're actually sacrificing themselves by releasing these things, possibly for the good of the rest of the population, to make these things and then cause some metamorphic effect in the animals that they interact with. And so what's the benefit? They're actually committing suicide, essentially, to make these things. What's the benefit to the bacteria? We don't know, but I think it'll be really cool to find out in the future. Okay, and so in summary, I think the future of phage tail-like structures and how they interact with animals is very bright. So I'd like to thank my advisor, Diane Newman at Caltech, my collaborators, Martin Pilhofer and João Medeiros, my funding sources, and I'll take any questions you might have. Thank you.